Our next presenter is Marc Fuchs. Marc was originally from France, where he received his master's and PhD degrees. And he's worked on viruses and for fruit crops and vegetables for the past 30 years. He's currently an associate professor with the Department of Plant Pathology and Plant Microbiology at Cornell University in Geneva, New York. Marc's going to be presenting this morning on the etiology of red blotch. Marc?
Anyway, I was asked to address the issue of the etiology of the virus, and rephrased. The question is the virus that has been isolated from red blotch disease vines the causal agent of the disease? And the starting point of our work was some extensive surveys that Sudi had done in numerous counties in California, including in Napa Valley. And here, I borrowed this slide from Sudi. Thank you, Sudi. Sudi surveyed about 12 different vineyards established with different cultivars and rootstock combinations, and tried to assess the correlation between symptomatic vines and the presence of the virus. And remarkably, Sudi's work indicated that a very high correlation between symptom expression and presence of the virus that had been isolated from diseased vines. And here the table shows a 96% correlation. However, this high correlation doesn't prove causality. So this is the issue that we try to address. And put in other words, we try to address and satisfy Koch's postulates. So what are Koch's postulates? Koch used to be, or was, a microbiologist in Germany, very much interested in human pathogens. And basically, he postulated that if one is interested in the causal relationship between a pathogen and a disease, that pathogen must be present in all of the cases where the disease is described. So Sudi's preliminary work satisfied that first postulate with a 96% correlation. Then second, the pathogen, if isolated from the disease host, needs to be somehow used as a pure culture when 3, inoculated to a healthy host, it would then induce the disease symptoms. And the last postulate is that the virus isolated from the newly infected host that becomes symptomatic has to be identical to the original inoculum. So we use 2 different approaches to try to fulfill Koch's postulates. The first one was to cure symptoms from infected vines. And the way we did it, we submitted Cabernet Franc that were infected with red blotch-associated virus to therapeutics treatments such as microshoot tip culture. And the population of plants that were produced after this therapeutics treatment were then carried over a couple of dormancy periods, after which they were tested for presence or absence of the virus. And we correlated presence or absence of the virus with symptom expression. And you can see here that 24% of the plants, or 13 of the 54, were asymptomatic and negative for the potential causal agent of the disease. And we were not able to remove this causal agent from 76% of the originally infected vines using this therapeutics method. However, we were able to cure symptoms using this virus elimination approach, which was encouraging in terms of the implication of red blotch-associated virus with the disease. Now, more importantly, we tried to reproduce symptoms using healthy vines. And there are different approaches that one can use to address this issue. First of all, grafting. Grafting, we would take a bud from an infected vine and chip bud it or top graft it onto a healthy vine. We decided not to do that although previous work at UC Davis had shown that red blotch-associated virus is graft transmissible, and the reason being that when one takes a bud from an infected vine in a vineyard, the likelihood that we are dealing with mixed infection is very high. So we might be dealing with a virus of interest, but also other viruses that might interfere with our experiments. Vector transmission is another elegant way to use a pure virus culture to prove causality. However, when the work was done, no vector of epidemiological importance was known for red blotch-associated virus. So we didn't pursue this avenue to address the issue of causality. The one we settled on was agroinoculation. So let me walk you through what agroinoculation is. Starting from the top left, from an infected vine showing typical symptoms like this Cabernet Franc, we isolated the virus, so we have a pure culture of red blotch-associated virus, and we transferred this virus into the bacterium called Agrobacterium tumefaciens, which is one of the bacterium associated with a Crohn gall disease of grapevines. And the reason we did this is Agrobacterium tumefaciens is designed to transfer genetic information into the grapevine that it infects. So the idea was using Agrobacterium as a vehicle to transfer red blotch-associated virus into healthy grapevines, and we use here healthy tissue culture grapevines, in other words, healthy vines propagated in tissue culture, with the idea that the bacterium will transfer the virus into these healthy vines that would then be observed to see whether we were able to reproduce red blotch symptoms. So basically, the way we did it, we just need three materials: the healthy plants that are maintained in tissue culture, the Agrobacterium strain that carries the genetic elements of red blotch-associated virus, and basically forceps and a little pin. And we dipped the pins into the Agrobacterium suspensions that were growing on solid medium, shown here on this Petri dish, and we basically pricked or poked the tissue of the healthy vines that were maintained in tissue culture. That was one of the options that we explored. The other option was to basically soak the healthy plants produced in tissue culture in a liquid suspension of the Agrobacterium carrying red blotch-associated virus. So regardless of the two options to deliver the virus via the bacterium, we observed the treated plants over time post-treatment to see whether we could observe red blotch symptoms. And this is sometimes what we had observed on Cabernet Franc, a treated plant on the left versus a control to the right, 1 or 2 months post-treatment. So we started to see some reddening, and underneath the leaf blade, the primary and secondary veins turned red. In Cabernet Franc, 2 to 3 months post-treatment, again, some of the leaf blades of treated plants turned red and like controls. What was interesting is the basal leaves of the treated plants had primarily expressed these disease symptoms that were very much similar to the red blotch symptoms observed in vineyards. However, as shown here, most of these basal symptomatic leaves desiccated very quickly and fell off the treated plants after 3 to 4 months post-treatment. So what we decided is, instead of carrying assays immediately, we again carried these plants through 1 or 2 dormancy periods to make sure that the virus had a chance to systematically infect plants, and that the virus was expressed from its own multiplication and not from the bacterium. And we tested also for the the bacterium in a subset of the population that we obtained, and we never detected the bacterium in treated plants after 2 dormancies. So again, in Cabernet Franc, the leaf on the left is showing what I would consider as typical red blotch symptoms, and like the control to your right. And we did this on different vinifera cultivars like Cabernet Sauvignon, Pinot Gris, and Chardonnay, where, as shown by Sudi in previous talk, we could see these sclerotic and sometimes necrotic areas of the leaf blade. The work was done also on rootstocks, and most of the rootstocks that were treated, like 110R, ripestris, or 3309C, some of them became infected with red blotch, but they remain symptomless. The only exception within the small group of rootstock genotypes that we have tested was SO4, for which an overall chlorosis of the leaf blades and a capping was observed in plants that became infected with red blotch. Now, the next step was to see whether the symptomatic vines did carry the virus. And this is just a subset of the plants that were tested here for the different genotypes of vinifera and rootstocks, and the number that we found infected with the virus in the second column. Now, I'd like to call your attention on the middle and the right-hand columns, and you see there a very good correlation between the presence of the virus, indicated by in the middle column, and the expression of symptoms on the right-hand column, indicating that symptomatic vines express symptoms only when the virus was present. And to fulfill the last postulate established by Koch centuries ago, we want to make sure that the virus that was present and detectable in the symptomatic vines was identical or similar to the inoculum that we used to start the experiments. And on a subset of plants that became symptomatic and infected, we sequenced the full-length genome of this virus progeny. And the results are very clear, indicating that what we recovered from the infected plants is identical to the inoculum that we used to infect them to start with. So in summary, our work has shown that symptomatic infected vines can be cured by microshoot tip culture. Agroinfiltrated vines can become infected with grapevine red blotch associated virus and exhibit disease symptoms. The symptoms are very similar, if not identical, on the infiltrated vines compared to the diseased vines in a vineyard. And the sequence of the virus progeny recovered from agroinfiltrated infected vines are identical to the infectious virus that was used as inoculum. And interestingly, and obviously of epidemiological importance, infection in rootstocks is latent in most of the genotypes tested. So just to conclude, based on these results, we can with confidence say that we fulfill Koch's postulates and reveal that grapevine red blotch associated virus is indeed the causal agent of the disease. And with no further ado, I'd like to acknowledge all the people in whether my lab or in Keith Perry's lab who have been instrumental in contributed to this research, and all the funding agencies, including all those who are currently attending and participating in the webinar. Thank you very much.